basic concepts of genetics. So let's start. So first of all, we have to understand the what is genetics. So here you can see the what is genetics. Basically, we comprise it into two groups where we can study the heredity and the variation. Heredity means what is the mechanism of inheritance, and variation means uh, how the variation has evolved by the means of sexual reproduction, or we can say by the means of mutation. Mutations is basically of two types: point mutation or gross mutations, and the sexual reproduction. Basically, in this mechanism, we have basically studying the crossing over. We talk about also the crossing over, recombination, and independent assortment. So before we going into further detail, let's talk about the father of genetics, which is known as Gregor John Mendel. So the birth of Gregor John Mendel in 1822, which born in Czech Republic, basically in the Brno city. There was a monastery where basically they work as a priest or monk, and it was a part-time job basically. And their study is basically from Vienna, and in the monastery they basically provided with a plot where they basically give a detailed study on the Physarum sativum. Basically, the common name of Physarum sativum is garden pea, where they did the experiment from 1856 to 1863. And this experiment, which basically did by the Gregor John Mendel, the conclusion of this experiment basically given in the 1865. Basically, the conclusion of this experiment was the concept of dominance and the segregation and independent assortment. Basically, they provided the three basic rules. The first rule, which basically we known as the concept of dominance, and the basically we considered the and second rule as segregation, and the third rule as independent assortment. So how Mendelism basically they conclude the results in 1865 was different from others. Why this happens? So basically the other scientists basically think that in the blending inheritance, it means mixed type of inheritance, and the Mendel basically they think that the particulate inheritance, um, actually the particulate inheritance basically you can see here, for every character basically every character has discrete particles basically, uh, for every character, for showing the every character they basically had a discrete particles or vectors which is responsible for that particular character, and it is a discrete factors basically, and which do not mix and are faithfully inherited from one generation to generation. It means Mendel says that the particulate inheritance, it means for every character for showing species has discrete factors which are basically responsible for that particular character. The other scientist below believes in the continuous variations. Continuous variations means just like we take an example of skin color, and we will we know that there are many types of skin coloration in the humans, and they believe that the other scientists believe in that mechanism. uh, but the Mendel was so practically basically they believe in the discontinuous variation Here you can see the graph phenotype just like that, from it a bell-shaped curve basically, which is the continuous variation. And there are so much variations, and the Mendel was says that the variation was a discontinuous variations. If we take a example of a plant height, so they said that the plant which will in the F1 generation will be in is tall, or if is dwarf. So there are there are two possible results were, one is tall or other is dwarf. There is no intermediate character showing in the F1 generation. So this is the belief that Mendel were think that. Now we talk about the rediscovery of Mendelian work, which is did by the Hughes, Carl Correns and von Teschmark in 1900. So basically frame the Mendel's conclusion from in form of laws. Basically, they give a conclusion of Mendel's experiment in form of laws. So just basically they repeated the Mendel's work and obtained the same results, and they give a conclusions. They give a concept of dominance. Basically, they give a law of segregation. They give a law of independent assortment. Uh, Basically, which work is done in pea plant, which is doing by the which is done by the Mendel, is also applicable on other organisms in most of cases. So basically the Mendelism was basically also applicable on other organisms in most of cases. So now we see the regions of failure of prior research. Why the prior researchers, basically the researchers who done their experiment before the Mendel was, why they fail? So there are so many reasons, but one of the most important is they think that they give a not pure line. Basically, uh, number one, parents heterogeneous, not pure line, variations and multiple locus phenotype. Basically, they take a heterogeneous type of parents, not which is not pure line basically, and they had a variation at multiple locus. So this is the first thing, and they investigate the multiple characters at one time, and which is so much confusion in that mind. So this is the basically first miss approach which basically fail that researchers, which basically failed the researcher. Second, the belief, basically they believe that the blending inheritance and Mendel was has basically only means, मतलब कि ये था कि जो मेंडल था वो ये सोचता था कि हाँ, जो कैरेक्टर्स होते हैं बेसिकली वो या तो अब मतलब ऐसा होगा कि अगर हम प्लांट की हाइट लें तो या तो वो टॉल होगा या फिर वो ड्वार्फ होगा. सो दिस इज द एक्सपेरिमेंटल अप्रोच. एंड बेसिकली जो रिसर्चर बेसिकली दे डिड नॉट यूज द एक्सपेरिमेंटल अप्रोच, थीमेटिक अप्रोच, and Mendel was the scientist which basically has the experimental approach or thematic approach. So there are so many reasons for Mendel's success here you can see, and from here we can understand the various introductory part of genetics. In the next lecture we will learn about the next reason for Mendel's success. So until from till now, say goodbye.